avian influenza. Okay, so my goals today are that you guys will be able to walk away from this presentation knowing lots of things. There's a lot of information here. I want to be able to go into lots of details. And so I did want to take the entire hour. Most of these slides have figures in them from like scientific publications, which will be cited along the bottom. So if you see anything that's of particular interest to you that you want to follow up on, the citations are at the very bottom of the slide. All right, so we're going to start today with influenza basics. What is influenza? Most of you guys are already familiar. You're going to know most of this stuff. We're going to cover it anyway. And then we're going to talk about avian influenza. How is it different from the influenza that we're used to? We're also going to talk about H5N1. So you've probably heard a lot about this lately. There's a lot of hubbub going on about H5N1, and we're going to talk about why. And then this entire presentation, I want us to think about this question. If we want to develop a surveillance pipeline focusing on avian influenza, what should we be focusing on? So a lot of this presentation you're going to find is very genomics based. So I'm going to give you the information that you need in order to understand how the influenza genomics space is working. But I want us to focus on this question as we go through the presentation. All right, so starting with influenza basics, this is something I'm sure all of you have seen many times before, but influenza is a virus and it causes seasonal epidemics in people. This causes flu-like symptoms. You've probably heard of this category of symptoms, including sore throat, cough, fever, body aches, headache, etc. A lot of these flu-like symptoms are shared with other infections that cause seasonal common colds, one of which is now COVID-19. But there are also other viruses that cause types of common colds, and these are most commonly caused by rhinoviruses, paramyxoviruses, and other non-COVID coronaviruses. So influenza is its own particular group and causes its own particular kind of set of symptoms. Influenza is primarily transmitted through the air via respiratory droplets, but it can also be transmitted via contaminated surfaces. And so you can catch it through direct contact with an infected person or by touching an object in the environment that has influenza virus on it. The best way to prevent infection is by avoiding these infected individuals, washing your hands and surfaces, and wearing a mask. And then in addition, you can also receive an influenza vaccine every year. So the influenza vaccine, vaccines provide immunity against influenza by priming our immune systems to recognize influenza by creating antibodies against the influenza proteins. And you also have some degree of natural immunity from previous exposures to influenza throughout your life. The not so great thing about influenza immunity is that the protection is usually incomplete. It's just the way our immune system works. It's not able to fully protect us against influenza. And one of the reasons this is that the major circulating strains of influenza change every year. And so influenza is constantly evolving across the world. And so your immune system will become adapted to the influenza from one year, but then it changes the next year. And so you lose some of that protection that you gained from your previous vaccinations and previous infections. And so that's why it's important to get your flu vaccine every single year, because these vaccines, essentially, they make educated guesses about which strains of flu are going to be the most prevalent next year. And so it's a way to kind of update that protection and protect you from the upcoming strains of influenza that they are hoping or thinking will be circulating in the following year. But that's also quite difficult. And so a lot of times, there are some times when we don't get it quite exactly right. And the influenza vaccine that's designed isn't a perfect match for what ends up circulating. So basically, the influenza vaccine is difficult. Also wanted to note that the influenza vaccine is made with inactivated viruses. So this is just an interesting tidbit, and it's different from, say, the COVID vaccine, many of which are based on these mRNA technologies. But influenza is the classic inactivated virus vaccine. All right, so now we're going to switch to avian influenza. So what is avian influenza? So the influenza virus, or the group of viruses that consist of avian influenza, also infect wild and domesticated birds. And these outbreaks can be extremely detrimental to poultry farms. Um, I want to make it very clear that avian influenza types are different from the types of influenza we get every season. But avian influenza can sometimes infect people too. And when it does, it typically has a pretty high mortality rate. But it is most common in people who have been exposed to infected poultry. And thus far, we have not seen outbreaks where it easily spreads person to person. So it is something that is pretty specific to birds. I also want to note that influenza is extremely diverse. There's a lot of different viruses in the influenza group, and they tend to be host specific. So, as I was mentioning, there is avian influenza that mostly infects birds. You've also probably heard of swine flu. And of course, we also have human flu. And these three groups of flu tend to be pretty distinct. And so, this is a phylogeny here on the right. You read this starting from the 12 o'clock position and reading it clockwise. And so what you'll notice is that the colors of these clades correspond to the host, which that influenza virus was found in. And you'll see that the bird groups in blue, the human groups in this orange-brown color, and the swine groups in pink, they are very distinct from one another. So they are circulating within their group, and they're not really being shared across groups very often. Another thing to note is that all of the known human strains of influenza that we get every single season are believed to be zoonotic, which means they come from animals. And we actually believe that most of these, in fact, probably all of them, came from birds originally. So, in fact, all influenza is avian influenza, but we now have some influenza strains that are adapted to humans and circulate specifically within humans. These avian influenza clades are also quite geographically structured. So you'll notice also here that some of these are labeled as Eurasian, and there's also this North American clade, well, just American clade. But these groups of avian influenza tend to be very geographically distinct. And that is because influenza actually circulates naturally in wild birds. And that is what is responsible for this degree of geographic structure. So, wild birds have these really nice north south migration patterns, and they tend to overlap in particular parts of the world. What you'll notice from this figure, though, is that there's very little overlap between Europe and Asia and North America. There are only specific small little regions in the Arctic where these migratory flyways overlap. And so that's why we tend to have quite a good distinction between the European and Asian avian influenza and the American influenzas. Um, and then there are, of course, the problems with increased industrialized farming, poultry farming, which generates lots of opportunities for these wild birds to come into contact with like very concentrated populations of like single species, which of course allows for outbreaks in these domesticated poultry farms. So we're going to switch gears a little bit. We're going to talk about genomics now. So the influenza genome is about 13,500 base pairs long. And the interesting thing about the influenza genome is that it is segmented. So the genome actually consists of 8 different pieces, and each of these pieces is a single strand of negative sense RNA. And each of these pieces contains a single gene. And all 8 pieces together code for 10 proteins. Most of these genes code for 1 protein, but there are 2 that are actually differentially spliced, and they actually code for 2 different proteins. So the entire virus is only made of 10 different proteins, 8 pieces of RNA and 10 proteins. So it's amazing that viruses are incredibly tiny and incredibly compact. Everything they need to do is encoded in these 8 small genes. The genes can further be split apart into categories. Some of these genes are structural and the rest are non-structural. So what I mean by structural is that these are the 3 proteins that essentially build the outside of the virus. They're the ones that actually make the housing for the rest of the viral pieces to exist inside. These non-structural genes are basically genes that replicate the viral RNA. So they are polymerase genes. And then there are some genes that coat the RNA and protect it. And so these we call non-structural proteins, and they exist inside the virus. When we talk about influenza, you're going to hear about H types and N types, and these refer to the hemagglutinin and neuraminidase proteins, the H type and the N type. So, for example, H1N1 or H7N9 or H5N1. And we do that because these are the proteins that interact with the immune system. So HA and NA are the proteins that stick out from the virus itself and interact with your cells. And so these are the ones that are attacked essentially by the immune system. And so we type influenza viruses according to these two proteins for that reason. I want to talk briefly about the life cycle of influenza. So when it infects your cell, what's actually happening. I don't want you to focus on any of these details really, but what I do want you to notice from this figure is that when the influenza virus enters the cell, it releases all 8 pieces of this genome into the cell. So now that these 8 segmented pieces are free, they are free floating within the cell and they are individually transcribed, translated. And when the virus comes back together, all 8 of those pieces have to come back together into a single virus. So all 8 of them have to return and basically be packaged into a new virus in order for it to continue replicating and go on and infect a new cell. Why do I want you to notice this? Because when you have a special case where 2 different influenza viruses infect the same cell, these segments can get mixed up. And this is called reassortment. So in this example, we have an influenza virus with red genes and one with blue genes. They infect the same cell, and now the red genes and the blue genes are both free-floating individually within the cell. And when a new virus gets packaged up, it can sometimes have combinations of both red and blue genes. And so this is what is called reassortment. And reassortment is really important for influenza viruses because it's a mechanism for them to rapidly evolve. So they can immediately go from having a particular red phenotype or blue phenotype and immediately build a new virus that has some combination of both red and blue phenotypes. And so this can allow them to evade the immune system or gain new combinations of traits. And this is something that is very specific to segmented viruses. So only viruses that have genomes in pieces can do this. And reassortment is important because we typically see initiation of new pandemics after reassortment events. And so this figure will explain from left to right the different pandemic influenzas that we've seen since 1918. So starting with 1918, the Spanish influenza that you've probably heard of. This was an H1N1 influenza virus, and we believe that this virus came directly from birds. And so starting with this H1N1 influenza that was circulating since 1918, what happened in 1957 is that this H1N1 human-adapted virus then reassorted with another avian influenza, in this case an H2N2 virus. And after this reassortment, we had a virus that had combinations of both the avian H2N2 and the human H1N1. And overall, it gained its H protein and its N protein from the avian. So this 1957 virus is an H2N2. Essentially, what we observed is that this then happened again. In 1968, there's another example where this reassortant H2N2 human-adapted virus reassorted with another avian influenza. And so now this 1968 virus is an H3N2, and it has some combination of both of the previous pandemic influenzas as well as a new avian version of influenza. And then this figure just illustrates that essentially this could happen again. We don't actually know. What I thought was really interesting about this paper is that it was published before 2009. Does anybody remember what happened in 2009? Yeah? No?
Swine flu.
Swine flu, yeah, exactly. So I thought it was quite interesting that they essentially, I mean, they were almost exactly right. What happened is that we had another reassortant pandemic flu, but this time it was a mix of the circulating H3N2 influenza, some avian influenza, and some swine influenza. So what we got was this mixture of all kinds of different influenzas, and they call it swine flu because this is the first time we've seen a pandemic influenza that has some of its gene segments from the influenza that are typically circulating only in pigs. And so again, I wanted to reiterate that we typically define influenza subtypes based on H and N. And so the swine flu was an H1N1, but these subtypes can have different patterns of reassortment among the other 6 segments. And so the other 6 segments are a combination of swine, bird, and human. Okay, we're going to talk now about these different subtypes a little bit more and where they circulate naturally. So, avian influenza viruses naturally circulate in wild birds, which we've already discussed. This actually includes 16 different H types and 9 different N types. So, in this figure here, everything in blue is influenza A, which is what we're talking about today. There are also influenza B, C, and D viruses. I've left this here just in case you're interested, but everything we're talking about today is influenza A. And so mainly influenza A circulates in wild birds. So I already said we have 16 H types and 9 N types. And then this circulation in wild birds can occasionally cause spillovers into domesticated poultry. So we've seen 13 different H types in domesticated poultry and all 9 N types. We can also occasionally get spillovers into humans, which have their own circulating types. So the 3 pandemic influenzas that I already mentioned, H1N1, H3N2, and H2N2. We have the circulating types in pigs, and occasionally we have incidental spillovers into other animals. Sometimes we have aquatic mammals, other wild birds, domesticated mammals, like cats and dogs. But generally, these things kind of keep to themselves. One thing that you'll notice on this figure is that there is a connection between humans and domesticated birds. And these are the avian influenza types that we're worried about that sometimes infect humans. And so the avian influenza viruses of H types 5, 6, 7, 9, and 10 have infected people in the past through exposure to infected poultry. But thus far, they have never gained this ability to circulate in humans continuously. So they are just incidental exposures and infections, not actually circulations. I wanted to add to this figure because now we have cows, which are not on this figure before. So we're seeing that this H5N1 virus, which we're pretty certain came from poultry, is now circulating in cows. And so we're going to get to this story a little bit more later. I want to talk about host tropism. So we're going to start to get into the weeds a little bit, but I'm going to try to simplify it down as far as I can. I want to talk about why avian influenza viruses don't infect humans, or why it's difficult for them to gain the ability to infect humans. And that's because when influenza infects a cell, it has to bind to a receptor. So there's something sticking out of your cell that the influenza virus attaches to, and that's how it gets into the cell. And the thing, essentially, that avian influenza or all influenzas attach to are these sialic acids that are sticking out of cells. So these glycans, they're called glycans because they're made of sugars, naturally exist on your cells. They're very important for different cellular functions, and this is what influenza takes advantage of. These sugars can be linked together in different ways, and we call these linkages alpha-2,3 or alpha-2,6. It doesn't really matter what that means, but what you should know is that alpha-2,6 is more common in humans and alpha-2,3 is more common in birds. And so the avian viruses in this figure are able to utilize these alpha-2,3 linkages, but not alpha-2,6. So in this figure, it's not sticking to the alpha-2,6, whereas the human virus is sticking to the alpha-2,6. And this slide just explains that a little bit further. So sialic acids are basically these final sugars that are linked to these glycans. These glycans exist on glycoproteins and glycolipids on your cells. And these terminal sialic acids can be linked in different ways, alpha-2,3 or alpha-2,6. And again, birds primarily have alpha-2,3. Humans do technically have a mixture, but we primarily have alpha-2,6 in our upper respiratory tracts, which is where influenza first infects. And so if it can't stick to those alpha-2,6s, it really struggles to establish an infection in humans. There are some alpha-2,3 linked sialic acids in the lower respiratory tract, but there is a mix of both 2,6 and 2,3. And then this figure also demonstrates the different distributions of these sialic acids in a couple different animals. And then the only thing I want to point out here is the ferret. So actually, ferrets are our best model organism for influenza because they have the most similar distribution of these sialic acids to humans. And so that's why you won't see influenza in a mouse model, because their distribution of sialic acid is quite different. And so you'll see ferret studies instead. And then if birds were in this figure, they would have an additional panel, and it would be mostly this light blue. So there's this very clear distinction between humans and birds. Okay, so now we're getting a little bit more into genetics. We're going to talk about why these H3 viruses and N2 viruses, H3 and H2, what happened in their genomes that allowed them to make this switch? Why was it that they were able to go from alpha 2.3 to alpha 2.6? And so what we have here are some alignments. We have an H3 and an H2 alignment here, and these are split. It's not very clear from this figure, so I highlighted it. The ones outside the box are avian, and the ones inside the box are human. And what I want to point out is that the human viruses unanimously have this substitution at position 226. So where you see a dot, it means that there is no difference essentially to the reference, and where you see a letter, it means that there is a difference. So all of the human viruses have this mutation essentially at position 226, which changes the amino acid from a Q to an L. There's also an additional amino acid that is unanimous among the human strains of influenza of this 228 position, and it changes from a G amino acid to an S. And so what we've figured out through some molecular studies and some epidemiological studies is that this single amino acid change at position 226 can allow the virus to actually switch from preferring alpha 2.3, which is avian, to preferring alpha 2.6, which is human. And this additional change at 228 actually enhances this preference. So I want you to remember these numbers, 226 and 228, for further in the presentation. We also have H1, like the swine flu. These adapted to mammals through slightly different pathways, so they have different positions of substitutions, but the important point to note is that there were still only about 2 changes required for these influenza viruses to go from preferring birds to preferring mammals, and humans specifically. And so the point of this slide is that you can get from birds to humans in as few as 2 mutations. So the types of avian influenza that we are most concerned about in humans are H5 and H7. So there have been incidences of infection for 6 and 9 and 10, but we are really only interested in H5 and H7 because these have caused the most frequent and significant outbreaks thus far. H5 and H7 are important because they can be further broken down into these 2 groups of highly pathogenic avian influenza, HPAI, or low pathogenicity avian influenza, LPAI. These designations are technically based on their mortality rates in domestic chickens, but this highly pathogenic phenotype is the result of basically the addition of an additional cleavage site in the H protein. Interestingly, this arose independently multiple times in the H5 and H7 group. But this cleavage site is considered a virulence factor in mammals. And so while this highly pathogenic designation is technically based on mortality rates in chickens, there is reason to believe that this highly pathogenic phenotype is probably also more dangerous for people. And so the H5N1 that's circulating in cows is actually a variant of highly pathogenic avian influenza. We're going to talk a little bit more about H5N1, where it started, and how we got to where we are today. So highly pathogenic H5N1 was first detected in domestic geese in China in 1996. And since then, it has spread across the globe in both wild and domesticated birds. And it's also reassorted with various N types, and as well as low pathogenic avian influenza strains. The H5 viruses have sporadically infected humans. Again, these people were only infected because they were exposed to infected domesticated poultry. But when they are infected, it does tend to have a concerningly high mortality rate. But also, just to reiterate, this does not transmit easily between people. Here's a timeline of the infections, the human infections of H5N1 that we have observed up until today, essentially. And most of these infections were in Europe and Asia. And you'll notice that there's a specific drop off in infections starting around 2017. And this is because this is when there was an avian influenza vaccination program implemented in domesticated birds. And so by vaccinating domesticated poultry, we can drastically reduce the exposure of poultry farmers to infected poultry and actually really, really inhibit these H5N1 outbreaks in people. I did say that H5N1 typically does not transmit easily between people, but one of the things that you should know if you're going to study avian influenza are these two landmark papers that were published in 2012. And so you've probably heard of these before. Maybe you're not specifically familiar with them, but these two studies are commonly pointed at as like examples of gain of function research. And when these two things came out, there was a lot of scrutiny and debate about the utility of gain of function research, whether this kind of thing should be allowed and how it should be monitored. And so we're going to talk about each of these in more detail. But both of these studies in 2012 wanted to understand what it would take for H5N1 to be able to make that switch to be able to transmit between humans, so to become transmissible through the air. And this, like I said, this prompted a year-long pause on all H5N1 transmission research. It was quite a big deal, and then initiated this increased scrutiny towards gain-of-function research. So we're going to start with the first paper. This is the Imai et al. Paper. And I want to, so you remember this 226, 228 position. So H5 seems to be able to similarly make this switch from alpha 26, from alpha 23 to alpha 26 using a similar pathway. So this 226, 228 pathway seems to also work for H5 in the same way that it works for H2 and H3. And so they started with this baseline. They have three different viruses here. They have a traditionally human virus, an avian virus, and then they have this virus that is an avian virus with the 226, 228 mutations worked into it. The red and the blue lines basically indicate how well this virus binds to human cells or how well it binds to avian cells. So if you see a panel that has a high blue line and a low red line, it means that that virus prefers birds. If you see the opposite, if you see a high red line and a low blue line, it means that virus prefers humans. And so you'll see that when you take the avian virus and you add in 226 and 228, it gains this red line ability to then infect humans. They also did some random mutagenesis screening, which means that they randomly inserted all kinds of different mutations into this influenza H protein and basically screened them to figure out which ones essentially could get this high red line phenotype, this ability to attach to the alpha 26 human-like sialic acids. And they identified one additional mutated H protein, and this one had 4 different substitutions in it. You will notice that one of them is 226, one of them is very close to 226, and these other two at positions 119 and 152 we haven't exactly heard of yet. And yes, again, this mutated variant appeared to be able to infect human alpha 26 linked sialic acids. Oh, some problems with my animations. So then they took this new variant that they had identified and they individually tested the combinations of these 4 different mutations that they had identified to see which ones were critically important. And they found that only 224 and 226 in combination can sufficiently make this virus able to infect with alpha 26 linked sialic acids. And that's this last panel here. The rest of the panels were either really weakly able to bind these human-like sialic acids, or they maintained their preference for avian sialic acids. What I want to point out and what they mention in their study is that binding in an assay does not mean natural infection, and it certainly does not mean that an infection can transmit between humans. So these assays are ways for us to assess whether a virus could bind to the human receptor, but it doesn't mean that if it did infect one person that it could then transmit to another person. And so they wanted to take their experiments one step further beyond these binding assays. And so they experimentally infected ferrets with these new variants that they had identified. And so they found that when they did infect ferrets, there were 2 new mutations that popped up that actually enabled this virus to gain the ability to transmit through the air. And so there were only 2 more steps essentially required for this virus to gain that ability to potentially be transmitted between humans or between mammals. We don't know humans because obviously we didn't do the experiments in humans. And so they're basically saying that they did this gain of function research, and they showed that there are very few steps that are required for this H5N1 to become transmissible through the air. And then switching to the second paper, this Herfst et al. Paper, they found the exact same thing with slightly different methodology. They started out with this 226, 228 mutation in H5 that enables the virus to link to these alpha-2,6 linked sialic acids. They also added this additional mutation in the polymerase gene, which enhances replication in mammals. We think this is because they have a different body temperature in birds. So they started with this virus that had 3 substitutions. And they found that these 3 substitutions were not enough for the virus to transmit through the air, which correlates with what we've seen in humans. These mutations allow it to infect mammals, but it does not allow it to transmit between mammals. So what they did is they did this study where they forced adaptation to ferrets by serially passaging nasal fluids of these H5 infected individuals. And so there's 2 rows here. They had one series of experiments where they used a wild type avian influenza. So this is an H5N1 without these mutations. And they also used an avian influenza that they added these mutations into. The numbering is slightly different here, but these correspond to 226, 228, as well as this 627 mutation in the polymerase. So they added these 3 things to the wild type avian influenza. And then they took each of these and they basically forced it to adapt to ferrets. So they infected the first ferret manually, allowed an infection to establish. They took nasal fluid from that ferret and then infected the next ferret. And they essentially did this 10 times for both viruses to see how the virus would change in ferrets as it was forced to transmit between them. And so then after 10 passages of this forced infection through the ferrets, they then had 2 different viruses, one that had adapted from the wild type and one that had adapted from the wild type with these 3 additional mutations. So after these 10 passages, they found that there was 1 new mutation that was detected in all the wild type sequences and in most of the mutated sequencing. And there was a nearby mutation in the remaining 11%. So basically, almost all of the strains had picked up this additional mutation in this 158 to 160 region. And it turns out both substitutions actually do the same thing. They removed a glycosylation site. And I wanted to point out that this 158D was actually found in the previous study. So both of these studies essentially discovered that this glycosylation site is important for airborne transmission. They weren't done, though. They took this 10th passage of this ferret-adapted strain for both the wild type and the type with the 3 mutations, and they inoculated it into new ferrets, and they placed these ferrets in cages next to recipient ferrets. So now, rather than forcing the ferrets to become infected by infecting them with nasal fluids, all they did was put them in cages next to each other and to see if the virus would naturally transmit in the air. And what they found was that only the ferrets that were in cages next to the mutated strain of avian influenza became infected. And then after they did this 3 times, they found one new mutation at this 103 position. And so the summary of this story is that you'll often hear that H5N1 is only 4 mutations away from a pandemic. And so the idea is that this like 226, 228 set of mutations can allow H5N1 to infect mammals and humans specifically. And then the addition of 2 additional mutations was sufficient for that virus to then be able to transmit between ferrets in these airborne transmission studies. So why are we so concerned about H5N1? And it's this in particular because it can already kind of pass this first barrier of 2 mutations in order to infect humans, and that they think that there's a really small barrier for that virus to then gain only a couple more mutations to allow it to then transmit between people. So there's this risk essentially that the virus could adapt to become airborne in people. So that's why we are concerned about the presence of H5N1 in dairy cattle. So we've never seen H5N1 circulating in cows before. This lineage was from a bird flu lineage that emerged in late 2020 in Europe. This virus was a reassortant virus between an H5N8 highly pathogenic avian influenza and an additional low pathogenicity avian influenza strain, both from wild birds. This novel lineage has caused record levels of infections in domesticated birds, and it's quickly spread all across the globe. And since then, it's reassorted a bunch of different times with other strains from wild birds. Excuse me. And this lineage and its reassortant progeny are important because they show unusually high propensity to be able to infect mammals. So we've seen this H5N1 avian lineage in domestic cats, wild and farmed foxes, farmed mink, wild seals. You may have read in the news that there's a H5N1 outbreak in pinnipeds at the moment, and also dairy cattle. This outbreak in dairy cattle is thus far constrained to the United States. Most of these infections have been found in Texas, but there are additional infection sites in other states. It's interesting that the infection sites in other states, such as the one in Michigan, appear to map to multiple different points on the phylogeny, which means that there were probably multiple independent introductions of H5N1 into Michigan, which indicates that this is probably circulating quite widely in dairy cattle. And the actual scope of the infection is probably much wider than this. I also want to point out on this phylogeny that there was in the news this incidence of the H5N1 infecting a dairy farm worker. But the genome of the dairy farm worker is actually quite different from the genome of the virus that they've been isolating from cows. And so it's not clear whether this dairy farm worker was infected from dairy cattle or whether he actually independently was infected by avian influenza from poultry. And it just so happens that it looks very similar to the one that infected the cows. Since then, we've also seen that this dairy cattle H5N1 is spilling over from cows into other species. And so you can look at this on the H5N1 GeoSeq dashboard if you're interested. These other species include chickens, cats, different types of wild birds, and different wild mammals. The important thing about this dairy cattle H5N1 is that there have been some amino acid substitutions observed in the polymerase gene, but actually none of the mutations in the H gene have been observed so far. So none of the 4 that would lead to adaptation to use the alpha 2,6 sialic acids for humans or to be able to transmit through the air between humans have been observed. So this 4 steps away kind of narrative doesn't apply here. This virus has none of those 4 steps yet. This doesn't mean that they won't slowly arise over time, but currently there's actually not a lot of cause for concern with this dairy cattle H5N1. They are concerned that this is like a new environment for flu. We haven't seen it circulating in cows before, so we don't know what's going to happen. But currently, there's no cause for alarm. And also, I wanted to mention that the FDA has pretty much conclusively announced that pasteurized milk is safe. And so there's no risk of becoming infected with avian influenza by drinking infected milk. The pasteurization process thoroughly inactivates the influenza, and so pasteurized milk is safe. Okay, we're going to switch now to talk about H7N9. So I did mention there were two types of avian influenza that we're most concerned about infecting humans. One is H5N1, and the other is H7N9. The H7N9 timeline of human infections is kind of similar to H5N1. We see 5 waves of epidemics between 2013 and 2017. But in 2017, this vaccination program was introduced into poultry. And since then, we've seen very few H7N9 infections in humans. This H7N9 virus was a mixture of H9N2 viruses and separate viruses that donated the H7 and the N9 genes. The mortality of the H7N9 infections in humans exceeds 30%, so it is quite high. The 226 mutation is already present in this H7N9 virus, as well as the mutation that we observed in the polymerase that allows it to adapt to the body temperature of mammals. These 2 mutations have been present in H7N9 since the beginning of the first wave. They've also noticed this G186V mutation that's also present. And then the highly pathogenic phenotype, so this cleavage site in the H protein, has also been present since the 5th wave. So all of these first 4 waves were actually low pathogenicity avian influenza, and only in the 5th wave did we see the HPAI phenotype. Also, according to this study, which ended in 2022, there were 0 cases in the last 3 years. And as far as I'm aware, there haven't been any cases since then. There have also been some ferret transmission studies with H7N9. So these H7N9 strains isolated from people do show some ability to transmit via respiratory droplets. There were 6 different ferret studies that were performed. The rates of contact animals becoming infected varied between 33 and 100%. Again, this 226 position is present in the virus that these studies were performed with, as well as this polymerase mutation and this G186V mutation. But what they found is that there's no consistent pattern in the substitution that arise after multiple passages between ferrets. So this is different from the H5N1 studies because they found that the same types of mutations were repeatedly popping up in H5N1, which leads to this like 4 steps away narrative. But it doesn't seem to be the same case for H7N9. There are substitutions that also cluster around this glycosylation site that is at positions 158 through 160, but there's not a consistent narrative in how H7N9 could adapt to become transmissible through the air. And so we tend to be a bit less concerned about it. And then I wanted to end with something a bit more hopeful and something I also find really, really interesting. If you're going to look up any of the papers from all of this presentation, I would recommend this one. It's very elegant and very interesting. I quite enjoy it. But basically, these scientists have found that we do have some existing protection against avian influenza H5N1 and H7N9 because there is some degree of cross immunity between these avian influenzas and the viruses that circulate in humans as seasonal flu. So we're going to talk about this concept of immunological imprinting. So with influenza, you have the strongest immunity against the first influenza type that you're ever infected with as a child. And these influenza types can be split into different groups. So these groups, there's 2 groups where the H types tend to be more similar to each other. And H1, H2, and H5 are in group 1, and H3 and H7 are in group 2. And so basically, anyone exposed to H1 and H2 as a child has some protection against H5. And anyone exposed to H3 as a child has some protection against H7. So there is some cross-reactive immunity here because of the exposures that you had to circulating influenza. This figure here on the right side, so these are the birth years across the x-axis, and they've also indicated the different years of the pandemic reassortant influenza outbreaks that occurred. So starting in 1918, anyone born in 1918 was most likely to be exposed to H1N1 first as a child. Starting in 1957, when H2N2, the pandemic started, anyone born in 1957 then has a slightly higher chance of being exposed to H2N2 first as a child. And so the figure at the top is kind of a simplification of the different strains that were circulating at different times. And across the bottom are their predictions of the proportion of children that were exposed to one virus versus the other according to their birth year. So you'll see that people born between 1918 and 1957 were most likely to be exposed to H1 first. Anyone born between 57 and 68 was most likely to be exposed to H2 first. And then anyone born after 1968 had a bit of a mixture of potential for being infected with H3 first as a child or H1. And what we notice with the bird flu infections of H7N9 and H5N1 is that each of these viruses tends to preferentially infect people of particular age groups. So these two figures across the top are the case counts according to the birth year of the person that was infected. You'll notice that H7N9 is preferentially infecting people with earlier birth years, so older people. And H5N1 is preferentially infecting people of more recent birth years, so younger people. And there's this really nice flip-flop, essentially, at 1968. So 1968 seems to be the breaking point between whether you're more likely to get H7N9 or H5N1. And the top two figures are the case counts, and the bottom two figures are the basically the excess mortality. And so we also see that mortality is more likely from H7N9 in this older age group, and maybe a bit more likely, but the picture's a bit less clear for H5N1. So if you're born before 1968, you seem to have some protection, some innate protection against H5N1. And if you're born after 1968, you have some innate protection against H7N9.